Hello everyone, welcome to the video on chemotherapy principles. In this video, I will explain about brief history of chemotherapy, principles involved in chemotherapy, and brief introduction about sulfonamides and Co-trimoxazole. This is my channel. If you like my channel, do subscribe. Now, Paul Ehrlich is considered as father of chemotherapy. The reason is he is the first one who developed chemotherapeutic drug known as Salvarsan. Salvarsan is chemically sulfonamide. This drug used to treat syphilis. In early 1900s, syphilis used to be a very dangerous disease, and to treat that, the first known established chemotherapeutic drug was Salvarsan, which was discovered by Paul Ehrlich. He received a Nobel Prize in 1908 for his anti-diphtheria serum. He is involved in developing microbiology, immunological products, and chemotherapeutic principles. Again, he coined a term called as magic bullet. Magic bullet means a chemical drug that kills the germs causing a specific disease without harming the rest of the body. And this concept has served the basic principle for chemotherapy. Now, let us see again about this magic bullet. Imagine, let's say someone has got a infection in a particular tissue. Inside the tissue, you have microorganisms are there. Now, what he meant by magic bullet is, by using particular drugs, the drugs selectively go and destroy this organism without affecting the surrounding tissue. So, the drugs will have selective toxicity against these germs or microorganisms without causing damage to this surrounding tissue. This is considered as magic bullet by him. Again, he coined certain terms like chemoreceptors. Chemoreceptors has formed the basis for chemotherapy. We'll see about them. But before getting into that, the basic definition of chemotherapy is this one. Understand each and every word. Use of drugs with selective toxicity against invading parasite. So this is what is broadly is chemotherapy is about. You will use drugs which have selective toxicity against invading parasites. Invading parasites means organisms which are attacking human beings. They could be external parasites, there could be internal parasites. External parasites come from the nature and they will attack human beings. They are bacteria, virus, fungi, protozoa and helminthes. Whereas we have an internal parasite known as cancer. Cancer is within the human body, and cancer is nothing but uncontrolled cell division. The divided cell will increase their mass and tumor, and this increase in mass and tumor will start attacking the surrounding tissue. So these are known as invading parasites. So in chemotherapy, we are going to read about drugs which are used to treat these parasites, whether it could be external or could be internal. So this is about invading parasites. And coming to selective toxicity, these drugs has got selective toxicity. It is because there are certain biochemical differences and differential sensitivity. Let us understand them in detail. Now, Paul Ehrlich also termed a word called as chemoreceptors. What he meant by chemoreceptor is species specific receptor. That means, think about this one. Bacteria has got cell wall. Now, human beings do not have this cell wall. So, use a drug which will inhibit cell wall, then it will be selectively binding to this cell wall and inhibit this cell wall without affecting humans. So cell wall becomes a chemoreceptor or species specific receptor. So he realized there is a difference in receptors in human beings and invading parasites. If you target that particular chemoreceptor, that will selectively affect invading parasite without adversely affecting human beings. So now this chemoreceptor concept is extended by these two principles. Now understand this, there are certain biochemical differences between humans and invading parasites, and that difference is nothing but one of the differences is cell wall. Similarly, there is a differential sensitivity is there. We'll see about these two principles. Now, biochemical difference, as I have explained, cell wall is found in bacteria but not in human beings. It has got peptidoglycan, and then you have cell membrane is there. So take for the case of penicillins. Now, penicillins are well-known established antibiotics. Penicillins selectively inhibit cell wall formation and they do not affect human beings. Why? Because there is a difference in biochemical structures. Human beings do not have the cell wall, they cannot attack it. That is what is one of the major principles of chemotherapy. Another one, look at this. This is a ribosome machinery. See, the basic functioning of ribosomes is very similar in humans as well as microorganisms. But the difference is in anatomy. In human beings, you have 80S ribosomes are there, which are made up of 40S and 60S. Whereas in bacteria, they have 70S ribosomes, which are made up of 30S and 50S. So there are certain drugs which will selectively go and bind with either this 30S or 50S. They cannot bind with 40 and 60S. So what happens? The drugs has selectively binding with this ribosomal apparatus of bacteria and inhibiting protein synthesis. And why this is occurring? Because of this biochemical difference. So this is what is meant by biochemical difference. Second one, differential sensitivity. Again, understand this one. Now, dihydrofolate reductase, it is also known as DHFR. This enzyme is responsible for the formation of tetrahydrofolate. This folic acid is essential for the formation of DNA. Now, this enzyme is present in bacteria, protozoa and mammalian cells. All of them has got this enzyme. But there are drugs which are selectively inhibiting only bacterial enzyme. For example, trimethoprim. Trimethoprim is 50,000 to 100,000 times selective towards bacterial DHFR, but it will not inhibit either protozoan or mammalian enzyme. So this is what is meant by differential sensitivity. Though the enzyme is same, human beings has got the enzyme, but these drugs are the bacterial enzyme is sensitive towards this particular drug, not human enzyme. Similarly, pyrimethamine selectively inhibits the protozoal dihydrofolate reductase enzyme, whereas methotrexate is selective towards mammalian enzyme. So there is a difference in their sensitivity, and that is what is known as differential sensitivity. So these two are basic principles on which chemotherapy resides. Next one. Now see, this is an example to explain both the principles. Now look at this. See, human beings will get folic acid from diet. So from diet, we all get dihydrofolic acid, which is converted to tetrahydrofolic acid with the help of this enzyme, dihydrofolate reductase. Whereas microorganisms or microbes have to synthesize folic acid from the beginning. So they take pyridine and PABA, synthesize dihydroterate, which is converted to dihydrofolic acid and finally tetrahydrofolic acid. So this enzyme is not present in human beings, and this is the biochemical difference. That's why sulfonamides inhibit this enzyme and block the biosynthesis of folic acid. So this is biochemical difference, whereas this enzyme is present in microbes as well as human beings, but there is a differential sensitivity. So to make you understand, I have given this chart. Understand this one. So biochemical difference is you have a particular cellular component which is not present in mammals, but it is present in microorganisms. It could be an enzyme or it could be cell wall. The other one, there is a differential sensitivity. That means though the enzyme is present, certain drugs are very selective towards microorganisms but not towards mammalian cells. So these two are the principles of chemotherapy. Moving further. Now let us understand about parasites. I told you these are external parasites. You know, we all know all bacteria, virus, fungi, protozoan, helminths will attack human cells and cause certain adverse effects. Coming to cancer. Cancer is internal parasite. Understand this diagram. See, majority of cancers are because of a random mutation. So certain cells will get mutated and they undergo hyperplasia. Hyperplasia means increase in number of cells. Look at here. The number of cells is increased here. After that they go to dysplasia. Dysplasia means there is no proper formation of cellular cells are there. So that condition is called as dysplasia. Now it results in a tumor mass that is known as cancer. Now once the tumor mass is there, because of its size, it starts attacking the surrounding tissue. See, all this tissue is broken down, is attacked because of this cancer cells, and that is called as invasive cancer. The surrounding tissues are attacked. Invading means attacking. Parasites, surrounding tissues will get affected because of these particular parasites. So cancer will attack the surrounding tissue, hence it is considered as an internal invading parasites. So these are all invading parasites towards which our chemotherapeutic drugs will be acting. So this is the introduction of chemotherapy. Now let us see about sulfonamides. Sulfonamides are antibacterial agents. They are first known antibacterial agents which are successfully used to treat bacterial infection. Now, this scheme I have already explained it. Let me explain it again. So, this thing. So, this is the same thing. Give me a second. Yeah. See, till here all microorganisms will synthesize folic acid, and these enzymes are present only in microorganisms, whereas human beings get this folic acid from the diet. Now, if you see the importance of folic acid, see, this enzyme is present in microorganisms. We don't have it. These two microorganisms. And finally, we get folic acid in this form, and it is converted to tetrahydrofolic acid. Now, the function of folic acid is this folic acid is converted to 5,10-methylene tetrahydrofolate. That means it is carrying a methylene group. What is the function of methylene group? See, in RNA you have uracil is there. DUMP means deoxyuridine monophosphate. That uracil is converted to thymine monophosphate. This with the help of an enzyme thymidylate synthase. Now, what is the difference when you see the structure between uracil and thymine? The difference is a methyl group, and that methyl group is donated by tetrahydrofolic acid. So, folic acid donates a methyl group and converts uracil to thymine, and that is what is required for DNA. If you remember, uracil is present only in RNA. In DNA, you have thymine, and that conversion occurs with the help of folic acid. This is what is the essentiality of folic acid. And in order to form new cells, you need DNA. In order to get DNA, you need thymine, and that forms with the help of folic acid and this enzyme. So, this is what is the significance of folic acid. Now, let us see it. See, bacteria will synthesize folic acid by combining pteridine with para-amino benzoic acid. PABA means para-amino. See, this is benzoic acid. To para position, ortho, meta and para position, you have amine is there. So, this is known as para-amino benzoic acid. So, this is how it starts synthesizing. It forms steroic acid, which combines with glutamate and forms folic acid and gets reduced. Now, sulfonamides. See, sulfonamide is this group. Compounds which has got this group is known as sulfonamides. If you remember, CONH2 is a simple amide group. In place of CO, if you have S double bond O, it becomes sulfonamide. So, drugs which contain this group are known as sulfonamides. This ring is known as sulfanilamide. Look at this. This portion is aniline. So, that is why you have anil is there. Sulfamide, sulfonamides. So, sulfanilamide is a chemical name for these molecules. Now, why it is showing activity means, if you see the structures, look at them, para-amino benzoic acid, sulfanilamide, both of them structurally very similar till here. What is the difference? Instead of benzoic acid, you have sulfonamide is there. Both of them sterically, if you see, the size is very similar. So, because the size is very similar, they go bind with this enzyme. In place of para-amino benzoic acid, the enzyme is bound with sulfanilamide, and the further progression will not occur. This is how they will act. They in fact inhibit this enzyme. They can be also considered as anti-metabolites because they are involving in this metabolism, inhibiting that metabolism, hence they can also be considered as anti-metabolites. Now, understand the structural activity relationship patterns. This one is a very good acid. pK is around 4.5. It easily donates H plus and acts as an acid. Now, sulfanilamide as such is not really good acid. You can increase the acidity by attaching electron withdrawing groups to this nitrogen. So, one of the hydrogen is replaced with electron withdrawing group, and it is replaced with electron withdrawing heterocyclic rings. Heterocyclic rings are very good electron withdrawing groups, and the effect is they increase the acidity, also they increase water solubility. If water solubility is increased, one of the adverse effect, crystalluria, is reduced. So, this is how most of the drugs of sulfonamide category are prepared. Look at them. So, sulfapyridine. See, look at this. This amine is considered as fourth one, this one is first one. So, all substitutions are made on this first one. A hydrogen is replaced with a heterocyclic ring. Here too, heterocyclic ring, heterocyclic ring, heterocyclic ring. Why? To increase acidity as well as to increase water solubility. This is what becomes structure activity relationship. Now, when you see the therapeutic uses, sulf-isoxazole is used to treat lower urinary tract infection and otitis media. It is an ear infection, To treat that, it is used along with erythromycin. Sulfadiazine. Toxoplasmosis is caused by Toxoplasma gondii. It is a unicellular parasite which causes fatal infection in pregnant women, and this can be treated with sulfadiazine. One more major use is there with sulfadiazine. It is used to treat burns. Next one, sulfadoxine is used to treat malaria. Look at this, prophylaxis and treatment. Before someone gets malaria, it is used for prophylaxis, and after getting it also, it is used with pyrimethamine to treat malaria. Sulfacetamide is usually used to treat ocular eye infections. Now the next one, sulfasalazine, is a combination of 5-amino-salicylic acid and sulfapyridine. Now this one will not get absorbed. It will not get absorbed. It passes till the colon. In the colon bacteria will release an enzyme called as aza reductase. With the help of this aza reductase, sulfasalazine releases 5-amino-salicylic acid and sulfapyridine. This 5-amino-salicylic acid is released at colon. So ulcerative colitis, itis means inflammation. The colon inflammation is treated by this drug, and this sulfapyridine is also used to treat rheumatoid arthritis. So this is specifically used to treat ulcerative colitis. So these are all the therapeutic uses of sulfonamides. Moving further, pharmacokinetics. See, in metabolism you have phase 1 and phase 2 are there. If you remember, by attaching a heterocyclic ring, you are increasing water solubility, so there is no need for phase 1. The major job of phase 1 is to increase water solubility. Already these drugs are water soluble. So what will be there? Phase 2. Phase 2 is conjugation reaction. They are acetylated. All the drugs are renally excreted. Some of the older drugs will cause crystal urea. The reason is when sulfonamides, when they have low water solubility, they combine together and form crystals. But newer drugs, there is an increased water solubility because of replacement of a hydrogen with heterocyclic rings. Again, they are highly protein binding ability is there. Especially in neonates, they may cause kernicterus. Kernicterus is caused when albumin will bound with bile pigments. But when sulfonamides are given, the bile pigment comes out and sulfonamides will go and bind with albumin. The free available of these bile pigments will cause a kind of neonatal jaundice known as kernicterus. Now the other side effects, see, all sulfur containing drugs will have this problem. They cause hypersensitivity. It ranges from rashes to Stevens-Johnson syndrome. Stevens-Johnson syndrome is a serious condition which needs immediate hospitalization. Next, again, all sulfur drugs, quinine derivatives, they may cause hemolysis in people with G6PD deficiency. Understand this one. Glucose 6-phosphate dehydrogenase. This enzyme, the job of this enzyme is they increases the levels of NADPH. The function of NADPH is to produce glutathione. The glutathione will protect blood cells from free radicals. Now understand the concept. If someone has got G6PD deficiency, NADPH formation is reduced, glutathione is reduced. So what happens? Free radicals will cause red blood cell damage, that is known as hemolysis. Understand this concept. Again, sulfur drugs will cause phototoxicity. So this is about pharmacokinetics and side effects of sulfonamides. Now the last one, trimethoprim. We have seen, see, the first step in the folic acid synthesis is inhibited by sulfonamides, whereas dihydrofolate reductase is inhibited by trimethoprim. Trimethoprim is very selective towards bacteria. This is what comes under differential sensitivity. Now usually the combination is given, trimethoprim and sulfamethoxazole, which is known as Co-trimoxazole. So usually the doses are in 1:5 ratio. That means if you give 20 mg of trimethoprim, 100 mg of sulfamethoxazole is given. This combination is used in neonates and children, whereas this combination is used in adults. To treat severe infections, a double strength, DS means double strength, which includes 160 mg of trimethoprim and 800 mg of sulfamethoxazole is given. Usually this is used to treat respiratory tract problems, tuberculosis. For tuberculosis also, second line drug combination is given in older days, but usually the respiratory problems can be cured. But the problem with this combination is it is not safe in pregnant women. It should not be given in pregnant women. So this is about Co-trimoxazole. Thank you for watching this video. If you like the video, do subscribe.